Okay, right. So this session is going to be recorded and also live streamed. So we are on Teams. Be careful what to say. Our first speaker for this session. So we've heard a lot this morning from Bristol people, and we're now going to hear from some of the people that we collaborate with. So I'm delighted to welcome Marc Gunter, who's going to talk to us about identification of causes of cancer through large-scale cohorts genetics and experiments.
Thanks. Got a pointer or anything?
There is.
Wow. Okay, so thanks for the invitation. It's really great to be here and to celebrate the I guess the end of ICEP 2 and moving into ICEP 3. Richard asked me to give a talk, and really what I'm aiming to do in this presentation is to really give some show you some new results. We've got some of our studies where we've been applying, for example, the proteomics within large-scale prospective cohorts, but then also to introduce you to some resources that we've been building over the last few years, some major grant-funded resources, which I think could be really relevant to some of the work that's being planned within ICEP 3 and might spark some ideas for further collaboration. Okay, so three main areas I want to tackle within the presentation. I'll see if I can get through all of them. But firstly, as I said, the application of proteomics within large-scale prospective cohorts to identify novel biomarkers and potential new causes of different cancers. And then secondly, multidisciplinary studies combining both exosomics, genetics, and experimental approaches to identify new causes and mechanisms of cancer development. And then thirdly, some potential for performing small-scale human intervention studies for investigating mechanisms of cancer development. I'll take my coffee. Just before I start, I don't know if any of you saw this, but there was the front page article in The Economist back in July that caught my eye when I was coming to the station somewhere in London, and I bought it. And I don't know if anyone was read it, but it's actually a really very nicely written article and very positive about all the developments and discoveries we've made in identifying causes of cancer and cancer prevention. So if you're ever feeling slightly despondent about progress that we're making within cancer research and particularly cancer prevention, I'd recommend reading this because it's actually really nicely written. And one of the journalists the journalist that wrote it, she was actually at the cancer prevention meeting in London in June. And I'd spoken to her and she actually quoted me in it, and she didn't tell me. So I was actually really surprised that suddenly you see my name. So luckily what I said was fairly sensible, but it's actually really nicely written. And Sarah Blagden and some of the other people who were at the conference also interviewed in there. So I'd really recommend reading it. But one thing it does highlight is the potential for the combining of epidemiology with experimental research and the power of some of these large-scale prospective biobanks now, where we can apply new technologies such as proteomics, metabolomics, genomics to these very large data sets where we have very detailed information on lifestyle exposures and medical history and then disease outcomes. And I think I don't need to tell you all about all the different cohorts that are out there. I know many of you have been using many of them, UK Biobank, EPIC, others for many years. But I think in terms of discoveries of causes of cancer, some might say that in terms of questionnaire-based data, we might have reached the limit of what we can discover around new causes of cancer. So obviously things like BMI, some aspects of diet, physical activity, occupational exposures. I think these cohorts have been really invaluable for affirming that these are likely causal risk factors for a number of different cancers. But perhaps that's as far as we can go with questionnaire-based data. And now we're moving into this era where there's a lot of new potential with molecular discoveries. And as I said, the application of molecular technologies to these very large biobanks and cohorts. And I think this really does open up opportunities for identifying biomarkers, not only of cancer risk, causal biomarkers, but also for early disease detection and novel drug targets. So I've been working on biomarkers and cancer risk for about 20 years now, and it's a field that's been full of frustrations and exciting discoveries. But I kind of reflected when preparing this about thinking, do we have many established biomarkers of cancer risk? And kind of looking at the literature and thinking about how to kind of subdivide some of these different biomarkers into whether they reflect biomarkers of risk or early detection and progression. I think it's fair to say that we actually have very few validated biomarkers of cancer risk. So think about etiologic markers. I mean, I think certainly inflammatory protein, C-reactive protein, IL-6, I think there's fairly consistent evidence that they're predictive of lung and colorectal cancer. Certain steroid hormones, for example, estrogens for breast and endometrial cancer. And then growth factors and related hormones such as insulin, IGF-1, which I think there's really quite consistent evidence that they're causally related to breast, endometrial and colorectal cancer. But beyond that, and I'm happy to hear anyone argue against it, I'm not sure if there are any other biomarkers out there, certainly circulating biomarkers, which we can actually say they're predictive of cancer risk and likely to be causally related to cancer development. And then we also have diagnostic or early detection biomarkers. So, for example, PSA for prostate cancer, KIM1 for kidney, CA125 for ovarian. But again, beyond those, we don't really have many good candidates. So I think the question we then can ask ourselves is, can the application of some of these new high-dimensional profiling techniques, such as proteomics, metabolomics, within large-scale prospective cohorts, can they uncover novel biomarkers of cancer and help us identify causes of cancer development? And I think the combination of prospective cohorts where we can measure biomarkers along with genetics and then working with experimental models, I think that is a really exciting path forward for identifying new causes of cancer and identifying biomarkers. So I think we're already making some progress in this area. So I want to show this example on IGF-1. So this is kind of one of my favorite hormones. I've been working with this again for almost 20 years, doing different studies in different cohorts, and often with quite conflicting results arising from different cohorts. So, for example, if you look at literature for IGF-1 and colorectal cancer, some of the early prospective cohorts, particularly those from the US, showed very strong positive associations between circulating IGF-1 and colorectal cancer. Over time, there was this kind of aggression towards the null. As the studies got bigger, and more and more studies measured IGF-1 in relation to incident colorectal cancer, we started to see relative risks which weren't significant or were much weaker. And I think there was some confusion in the field. That was all against a background of very strong experimental data showing that if you expose colorectal cells to high levels of IGF-1, they proliferate, they go crazy, and they can become cancerous. So we kind of knew from an experimental standpoint that IGF-1 is likely to be a causal role, playing a causal role in colorectal cancer development. But it really took the arrival of UK Biobank and having IGF-1 measured on 500,000 people to be able to arrive at more definitive evidence on the role of IGF-1 in relation to different cancer endpoints. So we looked at circulating IGF-1 in UK Biobank in relation to colorectal and breast cancer. And we also used a genetic instrument for IGF-1, which explains, I think, around 16% of circulating variation circulating levels, and applied that within GECKO and BCAC. And we saw quite clearly that there was this positive association between circulating IGF-1 and breast and colorectal cancer, both in UK Biobank, and then, albeit slightly weaker, in the genetic consortia. So I think this really demonstrates that putting together large-scale prospective cohorts where you have thousands of people with biological measurements combined with genetic approaches can help arrive at more definitive conclusions around the role of a particular biomarker and cancer risk. So what about new discoveries and identifying new biomarkers? So I'm going to talk a bit about some of the work we've been doing in EPIC, which I'm sure most of you are familiar with. It's a cohort of around 500,000 Europeans across 10 different countries who were recruited back in the mid to late 1990s. Around 350,000 had a baseline blood draw. and very detailed information on diet, lifestyle. And these people were median age around 51 at recruitment, so they're fairly old now. These people now in their 80s, 70s, 80s, 90s. The cohort's ongoing, and because of the size of the cohort and the length of follow-up, we now have a very large number of incident cases which have arisen since baseline. So at the last round of follow-up a few years ago, we had around 93,000 new cancer diagnoses which had occurred since baseline, including around 20,000 breast cancers, 10,000 colorectal, 9,000 prostate, and 2,000 kidney, and around 100,000 deaths. We're actually going through a new round of data centralization within EPIC now, and we're anticipating these numbers will increase quite substantially because of the age structure of the cohort and the length of follow-up. But within EPIC in particular, we have now been moving kind of away from more of the questionnaire-based, diet-based hypotheses, which were really the cornerstone of why EPIC was set up in the first place, looking at variation in diet and lifestyle across different European countries and then trying to link that to cancer, and really trying to put more resources into making it a resource for omics and biomarker studies in conjunction with questionnaire-based data. So EPIC isn't like UK Biobank, where they've measured lots of biomarkers on the whole cohort, because it was the way it's been funded is very different. So much of EPIC, much of the molecular data of EPIC have been generated through individual grants and generally nested case control studies. But we have been trying to develop new partnerships with industry. So I'm going to talk about some of the work we do with SomaLogic recently. We now have a new collaboration, hopefully with Regeneron, which looks like it's going to provide whole exome sequencing around 200,000 of the participants. But we do have a growing volume of omics data within the cohort. So we have around 40,000 participants with metabolomics, around 50,000 proteomics, and that's growing. And then just on the left, I can just get an example of colorectal cancer. You can see the kind of numbers we have for incident colorectal cancer cases. So quite fairly substantial numbers. So this study with SomaLogic, which we started a few years ago now, the idea was really to identify novel biomarkers of different chronic diseases, both etiologic biomarkers, but also markers of early detection of disease. So we established a case cohort in EPIC. So this was building upon the INTERACT study, which was a study of diabetes set up about 15, 20 years ago now by Nick Wareham. And we essentially have around currently 20,000 participants within the case cohort, where we have instant cases of different cancers, diabetes, cardiovascular disease, Parkinson's, and Alzheimer's disease, as well as deaths. And in conjunction with SomaLogic, we applied the SomaScan 7K assay, which measures more than 7,000 aptamers and around 6,300 unique proteins within a single blood sample. And the SomaScan assay, I think currently, obviously there are other competing technologies, Olink, some of the more mass spec-based approaches. But the SomaScan 7K, I think currently is kind of gives you the greatest breadth in terms of measuring different biological pathways, which we think might be relevant to disease development. So really spanning from the immune system and metabolic proteins through to non-secreted proteins, so proteins which are expressed by different tissues, cell adhesion proteins. We applied the 7K assay to around 20,000 EPIC participants. And then also we have a collaboration with the ARC cohort in the US, where they also have SomaScan 5K data on the whole cohort. So this has been serving as something of a validation cohort for us within the study. So just to give you a flavour of some of the results from this now. So this is obviously a very busy area. We have many people in EPIC currently working on different aspects of this analysis. But just to give an overview of currently where we are. This bar chart shows the number of different protein cancer associations for different cancers. Hopefully you can make some of them out. It's not very clear. And then the number of significant associations at FDR significance level. So we had around just under 2,000 protein cancer associations, which were significant at FDR level. by far the cancer with the greatest number of hits was liver cancer, so hepatocellular carcinoma, which had around almost 900 significant protein associations. But then also some of the other, particularly blood cancers, so CLL, multiple myeloma, then also some of the upper airway digestive tract cancers also had a large number of hits. But then you also can see that some cancers we actually detected very few associations. So, for example, for prostate cancer, ovarian cancer, postmenopausal breast cancer, we only saw generally less than 10 significant protein cancer associations. Okay, this is not very clear at all, but sorry about this. But essentially what we're showing here is for the different cancer types which have been evaluated, the top 10 hits for each cancer type by magnitude of association. So this is a pan-cancer analysis, which has been led by James Yomelinsky, who was here before, and Vivian Vialon. And although you can't really see very clearly, but we do see a lot of heterogeneity by cancer type. So, as I said, some cancers we see a lot of association. So this is liver cancer here, or HCC. Then others such as prostate, we see very few. So prostate, the strongest hit was PSA, which somewhat disappointing, but not surprising that comes up. In a way, that's a nice positive control that we've done the study right and set it up correctly. And then for other proteins, so other cancers, so for example, multiple myeloma, we see this very strong association with TNF17B, which is actually one of the drug targets for multiple myeloma treatment. So we do see a number of different protein cancer associations, which already been shown to be targets for drugs, which in a way is a good validation or affirmation that the study is telling us something that is already established. But we also discover lots of new protein cancer associations. So just want to give us a few examples before I go on to that, just to say we do see shared proteins across cancers. So there are around 60 proteins which had associations with at least 3 different cancer types, and around 12 which were associated with more than 4 cancer types. Many of these tend to be immune markers and inflammatory markers, which are associated with different types of blood cancers, but also with lung cancer and some of the upper airway digestive tract cancers. But also we see, for example, MUC16, which is also known as CA125. That comes up for ovarian cancer, which is already well established as being a risk factor, risk marker for ovarian cancer. But it also comes up for colorectal cancer and some other gastrointestinal cancers as well, which hasn't been so well known up until now. So what we are seeing is shared pathways, which seem to be proteins, which are common proteins, which seem to be shared across a number of different cancers, some of which have shared etiology. So, for example, we see a lot of the same proteins for some of the blood cancers, lung cancer, upper airway digestive cancers, which could potentially be linked to smoking or alcohol intake. So I think these sorts of approaches are helpful in picking up common but also distinct pathways for different cancer types. So just to give some more detailed examples for a couple of different cancers. So this is the results for colorectal cancer, which I know many people in this room are interested in. So on these volcano plots, on the left, we have total colorectal cancer, then in the middle, colon cancer, and on the right, rectal cancer. And we see, overall, we see around 60 proteins, which were significantly associated with overall colorectal cancer, FGR significance level. And these range really from very strong positive associations with TFF3, which is trefoil-like factor 3. Some of these proteins, so LAMA4, CRAC1, lectin mannose binding protein, these tend to be proteins which are expressed within colorectal tissue and play roles in cell adhesion. They form part of the extracellular matrix of the colorectal tissue. And then other proteins such as COMP, which has this very strong inverse association with colorectal cancer development. So COMP actually encodes the gene for collagen synthesis. And what's interesting here is COMP is actually a factor linked to prognosis of different cancers because it tends to reflect tissue remodeling particularly of colorectal tissue and breast cancer, and is actually a marker of poor prognosis. So it's interesting that it's coming up to be potentially protective, and I'll show in a moment that the genetic analyses also seem to suggest that it might have a causal role in colorectal cancer development. Some of these are known GWAS hits for colorectal cancer. So, for example, LMR4, CRAC1. What you see is also that we see a lot more for colon cancer. So some of this could be related to power. So we had overall we had about 1,000 colorectal cancer cases in this study, and around 600 of those were colon cancer, and about 300 rectal cancers. So we probably have less power for some of the rectal analyses, but nevertheless we do see some hits for rectal cancer, particularly some of the inflammatory proteins, which seem to be more relevant for rectal compared to colon cancer. And then you can see this 8-ODP protein here. So this is actually a protein involved in protection against DNA damage. Okay, so it protects against misincorporation of purine bases within DNA, and is a marker of tends to be a marker of DNA damage. And I think, I know it's interesting to speculate, but we know that rectal cancer has a stronger link with smoking and alcohol, maybe pro-oxidative exposures. So really interesting that this comes up for rectal cancer and not at all for colon cancer. As I said, we're very interested in trying to confirm these results. So obviously we can do what we can with an EPIC to try to limit false positive findings, but ultimately we want to try to confirm our findings in external cohorts. So within ARIC, they had around 270 incident colorectal cancer cases. Then UK Biobank, which has Olink data, not SomaScan currently, they have around 520. So we try to perform some kind of validation analysis within ARIC and UK Biobank, and we did see some consistency across the cohorts. So around half the proteins which showed significant associations in EPIC, we're also able to look at within the ARIC cohort. And we saw that around 10 to 15 of them also showed consistent associations, or at least directionally consistent, even if they weren't significant. Where things were a bit more tricky was when we actually came to do some of the genetics. So many of these proteins actually do not have good PQTLs or genetic instruments for them, because there haven't been good GWAS done on many of these proteins which are captured within these assays currently. So I think that will come. We're actually going to be part of a very large new GWAS on SomaScan protein. So I think after that, we'll probably have better genetic markers for some of these proteins. But for those that we could study, we actually did see evidence which was consistent with the observational evidence that COMP, TFF3 and INHBC seem to be playing a causal role in development or potentially playing a causal role in development of colorectal cancer. It's just interesting, TFF3 also comes up as the top hit for colorectal cancer in UK Biobank, measured on the Olink platform. And this is a protein which is expressed within gastrointestinal tract and plays a role in wound healing, and also interacts with inflammatory pathways. So it's potentially, because we see such a strong association with this, this could be a good biomarker of colorectal cancer development. Moving to talk about breast cancer. So again, breast cancer, we have very few established biomarkers beyond some of the steroid hormones and IGF-1. This is the data for premenopausal breast cancer, which again, we tend to have very few biomarkers and limited insight into the etiology of this cancer, even though it's becoming more common. We did actually see quite a large number of significant hits for premenopausal breast cancer. So on the left, we have the volcano plot for the proteins, and on the right, you see its forest plot, and the different color models that you can make it out indicate different types of adjustments we performed. So a basic model, and then adding different breast cancer risk factors, and essentially they didn't really seem to change the overall results. We also were able to replicate some of the findings in ARIC and UK Biobank. So the top hit, the one on the top right-hand corner, leg 1. So this finding really came out of nowhere. So we actually don't know what this protein does. It was cloned in zebrafish, and it stands for liver-enriched gene in zebrafish. So it doesn't really have any human cognate or known role in humans thus far. But it does come out as a very strong positive risk factor for premenopausal breast cancer. And likewise in ARIC, it was also their top hit for breast cancer. Other proteins, so LACRT, so this is actually a gene protein which is involved in essentially initially identified in tear ducts, but is expressed within breast tissue. But again, we have no real understanding of what it might be doing in terms of its biological function. AGP glucokinase, that's the metabolic enzyme, which has been linked to a number of different cancer types. And then CXCL13, so this is a chemokine, which has been linked to breast and other cancers previously, playing a very important role in the inflammatory response. So some really interesting hits here, I think. I mean, particularly this leg-1 hit, which we also found for, again, breast cancer, for postmenopausal breast cancer, comes up with a very strong positive risk factor. We saw far fewer hits for postmenopausal breast cancer compared to premenopausal breast cancer, which actually was quite surprising. And then again, tried to confirm the results in UK Biobank and in ARIC. So UK Biobank, where we have O-link data. They also have leg-1 measured on the O-link assay, but we didn't really see the results null. This is for postmenopausal breast cancer, whereas we saw a strong positive association in EPIC. But for leg-1, CST6 and LAC1, we were actually able to confirm those in ARIC and in UK Biobank, which had some confidence that we are picking up something meaningful. Again, we really struggled with some of the genetic analyses. So we're only able to evaluate 20 proteins out of all those which were significantly associated with breast cancer. And that's again because of lack of PQTLs and genetic instruments. And again, the results were fairly kind of all over the place. I think the one exception was leptin, which we saw consistent evidence from both genetic and observational data. So that just really gives a snapshot into some of the findings we're getting from this. I do think the application of some of these new technologies, such as proteomics to cohorts, has the potential to uncover novel biomarkers and biology, and might help us identify new targets for prevention, detection of biomarkers. I haven't talked about some of those detection work that's going on, but I do think there's a lot of promise there. We're also doing some work with risk prediction models and also integration of blood with tissue-based biomarkers. But I really want to stress the kind of importance to follow up some of these with experimental and genetic analyses, really for triangulation of evidence. And I think I know that's a key feature of some of the work that's going to be going on in ICEP. Also, just to say, this data are available and can be used for testing specific hypotheses. So this is the work that James led and just published in JNCI a couple of months ago, looking at different lipid-lowering drugs in relation to cancer, where he found that genetically proxied ANGPTL4 inhibition lowered risk of colorectal cancer by around 25%. And he demonstrated that genetically within gecko. And also working with tissue-based data, found that loss of function variants associated with reduction in proliferation and Wnt signaling in colon tissue, again, consistent with a causal effect of this gene. But we actually decided to see whether this was on the SomaScan platform, and it actually is. So there is measurements of ANGPTL4 within the SomaScan 7K assay. And we looked at that in relation to colorectal cancer in EPIC and found consistent results, what we see from both the genetic data and also the experimental model. So again, we're able to triangulate different types of approaches to arrive at likely causal effect of this protein on colorectal cancer development. So just to say, if you have particular proteins or targets of interest where you think having observational proteomics data might also be useful, do get in touch, because I think it's a really great resource for, again, for this type of approach. So just moving on now to some kind of multidisciplinary work that we've been doing to identify new causes of cancer. So we'll talk a bit about Discern, which is a European Union-funded project that I co-lead with Paul Brennan at IOC, which is aiming to uncover new causes of 3 different cancers: pancreatic, kidney, and colorectal cancer, using a combination of epidemiology as well as experimental approaches. So this is a very multi-partner, multidisciplinary project. We're about halfway through the 5-year project now. These are partners on the right, and this was us at our recent retreat in Brno in the Czech Republic, where we had our annual meeting. And it's actually been really, really fun and exciting project to work with many people that we hadn't worked with previously, but also working with lab scientists who often speak a very different language to epidemiologists, and trying to kind of arrive at common understanding and thinking about what we're doing, and trying to make sense of each other's approaches has actually been really, really interesting. So the rationale for Discern was really trying to understand variation incidence rates of these 3 different cancers across Europe. So if we look at kidney, pancreatic, and colorectal cancer, we can see there is some marked variation in incidence rates in different European countries. So particularly for kidney and pancreas, we see this kind of hotspots of high incidence in many Eastern and Central European countries. So for kidney, very high rates in Czech Republic, Slovakia, Latvia, Estonia, but comparatively lower rates in some of the southeastern European countries. If you look at pancreas, again, we see the highest rates in Hungary, Czech Republic, Austria, Estonia, and then much lower rates in, for example, UK, Spain, Portugal. And again, for colorectal cancer, it's again very different. So highest rates are actually in Norway. So Norwegian women have one of the highest rates of colon cancer in the world, much higher than in Sweden or Finland. But Denmark, Netherlands also have very high rates, Hungary, and then comparatively low rates in Finland, Germany, Austria, Switzerland. So, I mean, one thing I should point out, these are not kind of like kind of global variation we see in some cancers. So, although they are different, they're not drastically different. As, for example, comparing rates in Africa with those in Europe or North America, but still there is significant variation in incidence across European countries, and it has been speculated this could be caused by particularly environmental exposures. So we have colleagues in Norway who are desperately trying to understand why they have the highest rates of colon cancer in Europe and also in the world, and why they should be so different from their neighbours in Sweden or Finland. So is it pointing towards a particular environmental exposure or something, for example, in their diet, that could be driving colon cancer in this population? And in terms of what we know about these different cancers, so we can explain, particularly for kidney and pancreatic cancer, around 30% of the attributable risk from obesity, tobacco, and then for colorectal, around 50% again from obesity, alcohol consumption, tobacco, various aspects of the diet. But there's still a lot that we don't really understand. And I guess some of the rationale behind Discern is, can we exploit some of these new technologies to uncover novel causes of these cancers and help try to explain some of this kind of missing, this gap in our knowledge about causes of these different cancers. So some of the key questions we're trying to address in Discern are, firstly, can we understand why cancer incidence varies between different regions and countries? Secondly, can we identify new causes of these cancers using combination of population-based and experimental research? And then thirdly, can we identify mechanisms, underlying risk factor cancer associations? And in particular, we're interested in the effect of cancer risk factors on normal tissues that can help us understand mechanisms and early origins of cancer development. So I don't have time to go into all the details, but essentially Discern is broken down to 5 main objectives. So the first objective is really trying to pull together a very large biorepository from European cohorts. In the second objective, we use these data and these samples to apply exposome type approaches, so metabolomics and proteomics within these cohorts to identify proteomic and metabolic risk factors for these different cancers. And then in objective 3 and 4, we take this through to the laboratory. So findings from some of the epidemiological studies, we then test whether, if we identify, for example, a particular compound from the human studies, which seems to be associated with a pancreatic cancer, take that into experimental models within the lab and see what's it doing to pancreatic organoids, for example. Can it help us understand mechanistically how if that particular compound is associated or is playing a role in that in pancreatic cancer development. And then the fifth objective is really about dissemination and trying to influence public health policy. So this is funded as part of the EU cancer mission. So to try to really drive understanding of what's causing cancer in Europe. So an important aspect of this is trying to have influence eventually on, if we do identify, for example, an environmental cause of some cancers in a part of Europe, to actually use this evidence to do something about it at the parliamentary level. So just to give an overview, because I think some of this could be relevant to OCEP going forward, because I know discern is going to be part of some of the objectives of OCEP. So building this sort of biorepository of cohorts and tumor tissue. So we've tried to capture as many cohorts in Europe as possible, and in particular try to include countries that show high rates of some of these different cancers. So it's actually quite challenging because in Eastern Europe, we don't have large prospective cohorts like we have for EPIC, like we have in Western European countries. So there are some small cohorts in Eastern Europe. So the PONS cohort in Poland, Estonian Biobank, which is actually quite a large cohort, and then the HAPPY cohort, which no one's ever heard of, but it's actually a very nice cohort set up in Lithuania, Poland, and Czech Republic and Russia, but they've been kicked out of it. It's actually a nice resource. It's really been underutilized for cancer research. So bringing in these Eastern European cohorts in, along with EPIC, and some of the other Western European countries, so NOAC, which is a large Norwegian cohort, part of which is in EPIC, but not in its entirety, in the form of a large biorepository. We also have tumor case series, so an important part of this study is not just working on blood samples and cohorts, but also using tumor samples, which have been collected where we have, for example, whole genome sequencing and other multi-omics, which have been performed on the samples. So again, trying to capture countries which have high and low rates of these different cancers. So for example, we're making use of samples which were collected as part of the Nutagraph's Grand Challenge. So we have samples of kidney, pancreatic, and colon samples from many of the Eastern European countries, but also collecting new tissue samples. So for example, in Italy, which has comparatively low rates of colorectal cancer, for example, we've just undertaken a new collection of colorectal tumours and normal colon tissue. And also in Spain, which has seen quite substantial increases in colorectal cancer in the past 10 to 15 years or so. We've collected new colorectal biopsies in Spain. So these all undergo whole genome sequencing and other multi-omics measurements. Then, as I said, in objective 2, we are going to be firstly performing what we call an external exposome analysis. So using geocoding and exposome surfaces, where we can try to identify, are there correlations between cancer incidence and particular environmental exposures. So using exposome surfaces, which have been collected in around 1 million Europeans within these cohorts. And this really goes from physicochemical exposures, built environment, for example, noise pollution, light pollution, food and lifestyle, and also social deprivation. And really kind of used in some of these maps which have been generated, which show if you look at some particular exposures, you do see some hotspots of exposure. So, for example, for PM 2.5, very high levels in some Eastern European countries. So really interesting, for example, Czech Republic and Hungary have very high exposure to some of these compounds. And that's where we're seeing some of the high rates of some of these cancers, particularly kidney cancer and pancreatic cancer. And then using the blood samples from these cohorts, we're performing endogenous measurements. So using both liquid and gas chromatography mass spectrometry, and hoping to cover as much of the internal exposome as possible. So capturing, for example, pesticides, drugs, surfactants, different complexing agents, hydrocarbons. So we have very kind of really wealth of metabolomics data within these cohorts, plus proteomics on a large proportion of the participants. And then, as I said, in objective 3 and 4, really trying to translate these findings into the laboratory. So this is mainly led by a group in Leiden in the Netherlands, where, for example, using organoids and different cellular models and exposing these to some of the compounds, for which have already been linked to cancer, for which we don't really have understanding mechanisms. So, for example, aldehyde acid or some of the PFAS compounds, but also identifying, using some of the results from the epidemiological analyses from objective 1 and 2, kind of feeding those into the laboratory. And the lab has already been quite busy trying to kind of start some of this work. So they've done some work, for example, on kidney organoids, exposing the kidney organoids to aldehyde acid, which has a very strong mutagenic signature for kidney cancer, and seeing some really quite interesting results in terms of some of the proteins which have been expressed within these organoids when you expose them to this particular compound. So, for example, GDF15, which is a protein which comes up as being associated with everything in some of the proteomics work, very long hit for exposure to aldehyde acid. So really the idea of discern, firstly applying these technologies, I think a really important component of this is some of the computational work that needs to go on, because we're going to be generating all this omics data from cohorts and also experimental models, trying to think about how to put some of that together and using kind of machine learning approaches, for example. And ultimately, what we're trying to do is identify new causes of cancer as well as causal mechanisms, which could then be used to inform prevention strategies. So, as I said, we're about halfway through the project. We're already generating quite a lot of data. By the end of this year, we should have a lot of the metabolomics data generated. But I think it'll be good if anyone's interested in this particular resource and wants to collaborate. We're very happy to work with you. So, in 2 minutes, just to quickly say, I'm also very interested in small-scale human intervention studies as a way of investigating mechanisms of cancer development. So I've been doing some work looking at diet-induced weight loss and collecting human tissue samples pre and post weight loss and applying molecular technologies to identify what actually happens in some of these tissues when we remove exposure to particular cancer risk factors such as obesity. So I'm part of one of the cancer band challenges, so prominent, which is trying to identify or understand how cells and tissues maintain normal phenotypes while harboring oncogenic mutations and how they transition to becoming a tumor. So I'm sure many of you know, but the conceptual idea behind prominent really came from 2 main observations. Firstly, that if you look at normal tissues, they contain a very high frequency of cells with known driving mutations, and this tends to increase with age. So even though the tissue remains healthy and not cancerous, we know that there is a lot of cells with known cancer driving mutations within those tissues. And secondly, really an observation coming from the mutagraphs is that many known carcinogens are not mutagenic. So it really does kind of raise the hypothesis, which has been around for some time, is that many carcinogens are actually acting in a different capacity, so actually acting as promoters. So in this model, what we see is that we have cells which contain or carry these dormant driving mutations, and they remain dormant until this promotion step, which many carcinogens might be acting as, so for example, some pesticides, some pollutants, obesity, diet, That's the second step, which is required then for malignant transformation. And because potentially this is modifiable, there's a lot of interest in this from a cancer prevention perspective. We can identify how these carcinogens are acting mechanistically. Is there a way then to intervene in this step or to prevent it occurring? So within Prominent, it's very multidisciplinary complex project. So Paul is leading a lot of the work, human samples, building upon the resource that was collected within mutagraphs. I'm doing intervention studies where we're collecting normal tissue samples and going through these interventions. Then in parallel, we have mouse models, Alan Balmain's group in California, who are performing many analogous interventions to what I'm doing with humans, but he's doing it within mouse models. Calvin Liu's doing a lot of organoid work. And then Nuria Lopez-Bigas really leading in much of the sequencing and computational aspect of the project. So in terms of the intervention work, what we're doing is collecting sequential tissue samples from people, non-cancer patients, healthy individuals, where we want to investigate the impact of removing exposure to a particular cancer risk factor and what is the impact of that on normal tissue biology. So 3 interventions: so intentional weight loss, which we're achieving through bariatric surgery, smoking cessation, and then chemotherapy treatment. And the idea is to collect normal tissue biopsies at the time of recruitment, and then roughly 9 to 12 months after the initial sample collection. And then these samples will undergo various molecular measurements, so bulk RNA-seq, single-cell profiling, spatial proteomics. Will also be derived organoids from these samples. So for the bariatric intervention, which I think is most applicable to some of the work in ICEP, we are almost at the end now of collecting these samples. So we've currently recruited around, we have about 113 baseline endometrial samples, 104 breast and 122 colorectal biopsies. So these have been collected both at Imperial and in Manchester. And already many of the participants have come back for their second biopsy. So we have around 49 endometrial, 29 breast and 41 colorectal. And we're aiming to get the full set completed before the end of this year. We are also exploring the possibility to add a GLP-1 receptor agonist arm to this, because we already set up an infrastructure for the bariatric work. Because of this interest in some of these new weight loss drugs, it'll be really interesting to see how does the effect of GLP-1 induced weight loss compare with bariatric surgery. These are just an example of some of the tissue samples that we're collecting within the intervention. So different tissue biopsies, fecal samples, breast fat, bloods, buccal swab, mouthwash, and cervical smears. So very large biorepository, very unique, which we're collecting within these interventions. As I said, all these samples will undergo bulk RNA-seq and spatial proteomics and also sequencing in the next year to 18 months. So this will be a very large data set which is available for analysis. But also we now have the biorepository. So if you have particular interest in these samples or ideas of what you'd like to do with them, particular technologies you'd like to apply to the samples, I'm very happy to talk about collaboration. Maybe I'll skip this because I'm running out of time. But just to say, I've been doing some work with Emma Vincent and Emma Hazelwood, who's on here. And we've already done some work using samples from intervention studies to identify potential causal proteins linking obesity and weight loss with colorectal cancer, and combining both data from interventions, but also some tissue-based work and also some knockout models that Emma's been running in her lab. So I think already here we have some proof of principle that combining these human interventions with experimental approaches can help us understand potential new causal mechanisms. Just there's potential with they can provide insights and etiology factors. I really just stress we need multidisciplinary teams, because we're working with epidemiologists, lab scientists, and really good clinicians. So I think that's a lesson that I've learned from some of this work that I've been doing. None of us can arrive at some of these conclusions working just within our own discipline. So I think we really need to kind of work together with people from different avenues of research. And I think also a question that's always on my mind is how to translate some of these findings into prevention. So all this discovery work, then what? Obviously, there's a whole interest in identifying drug targets, but that's not always as straightforward as it might seem. So I think it'd be good to have some more discussion about actually how to kind of move some of these discoveries and translate them into effective cancer prevention strategies. Okay, and thanks to many people, which I've probably missed some of them, but yeah, thank you very much.